I want you to imagine, just for a second, that you are sitting in a sterile, brightly lit doctor's office.
Right, a situation nobody really wants to be in.
Exactly. You've just received news that completely shifts the ground underneath you, a diagnosis of prostate cancer.
It's terrifying. And once that initial shock settles, the very first question is usually, well, how do we treat this?
Right, but to answer that seemingly simple question, your oncologist needs a highly specific piece of information. They need the genetic signature.
Yeah, the unique molecular fingerprint of your specific tumor.
And the problem is that this vital, potentially life-saving information is currently locked behind a massive systemic bottleneck.
It really is. To get that genetic read, doctors have to rely on proprietary, expensive molecular tests.
Which, requires physically destroying a piece of the biopsy tissue they just took out of you.
Yeah, you literally have to grind it up.
Right, and it takes weeks to process. And crucially, if you happen to live outside a major, well-funded medical hub,
you might not have access to this test at all, which creates a severe equity issue in modern medicine.
So true. You have the map, but you aren't allowed to read it. But what if we didn't need that expensive test at all?
That's the big question.
What if an artificial intelligence could be trained to look at a standard century-old glass microscope slide, the kind that cost pennies to make and is sitting in literally every hospital pathology lab on Earth?
And decipher that invisible genetic code just by looking at the shapes of the cells.
Exactly. And that brings us to the source material for this deep dive. We are looking at a groundbreaking March 2026 study published in Nature's NPJ Precision Oncology.
Authored by researchers at Northwestern University and the University of Minnesota.
Okay, let's unpack this. Because whether you are deeply invested in tech, a medical professional, or just an intensely curious learner, what we are exploring today is an absolute masterclass.
It really is. It shows how artificial intelligence might finally democratize precision medicine.
So to really grasp the magnitude of this breakthrough, we have to start with the biology, right? We need to understand what the AI is actually trying to find.
Yeah, exactly. In oncology, there's a concept called molecular subtyping.
Right.
We used to think of a tumor as just a rogue lump of cells. But we now know it operates more like a highly complex, corrupted machine.
And depending on the underlying genetics, like the RNA blueprints driving that machine, it operates very differently.
Precisely. So for prostate cancer, the medical field relies heavily on two specific classification systems to group these machines into categories based on their RNA or their transcriptomics.
Meaning we are categorizing them by the instructions the tumor is reading to build itself?
That is a great way to put it, yeah. The first system you need to know is called PAM50.
PAM50.
Interestingly enough, this system was originally developed to classify breast cancer.
Oh, really?
Yeah, but researchers discovered it works incredibly well for prostate cancer, too. PAM50 divides these tumors into three main families. You've got basal, luminal A, and luminal B.
So if you picture the structure of a prostate gland, you can think of the basal cells as the tough foundational scaffolding cells on the outside.
Right.
While the luminal cells are the specialized ones lining the inside of the ducts.
Exactly. So that's the first system, sorting them by their structural origin.
And what's the second system?
The second system is called the PSC, or the Prostate Subtyping Classifier.
Okay, PSC.
Unlike PAM50, the PSC was built from the ground up specifically for prostate cancer. So it gets significantly more granular.
How so?
Well, it categorizes tumors into four distinct types: basal immune, basal neuroendocrine, luminal differentiated, and luminal proliferating.
Honestly, for someone sitting in that doctor's office, hearing all these scientific classifications might just sound like alphabet soup.
Oh, absolutely. It's overwhelming.
So why does it actually matter if your tumor falls into a basal neuroendocrine bucket versus a luminal differentiated bucket?
Because it dictates survival. It matters entirely because of how these different subtypes react to our primary weapons against the disease. Specifically, prostate cancer is highly dependent on testosterone.
So doctors use androgen axis-targeted therapies.
Which is just the clinical term for hormone therapies designed to shut down the body's testosterone production.
Because the prostate cancer often feeds on that hormone.
Exactly. But here's the catch. Different molecular subtypes respond completely differently when you suddenly cut off their fuel supply.
Wow, okay.
Some subtypes will immediately starve and die off. Others have genetic workarounds. They just ignore the lack of testosterone and keep growing anyway.
So knowing a patient's exact subtype tells the oncologist whether hormone therapy will be a silver bullet or a complete waste of precious time.
Spot on. But getting that subtype brings us right back to the bottleneck we talked about.
The testing.
Right. To get those PM50 or PSC classifications right now, a doctor has to order what's called a deciphered gene expression assay.
And from what I understand of the process, they literally take the highest grade, most dangerous looking piece of your physical biopsy.
Grind it up.
Yeah, grind it up and run a complex genomic sequencing analysis on it.
Which consumes that precious tissue forever. Once you grind it up for the Decipher test, you can't go back and look at the cellular structure again.
And it's not cheap either.
Not at all. It costs thousands of dollars, and it limits global health access because you need highly specialized sequencing labs to run it.
So the researchers behind our source material wanted to circumvent this entirely.
Yeah, they proposed a radical shortcut. They wanted to see if an AI could predict the exact results of that expensive Decipher test by just looking at standard H&E stained biopsy slides.
I want to pause on the H&E slides because this is where the contrast gets wild. H&E stands for hematoxylin and eosin.
Which is not new tech at all.
No. Pathologists have been using these pink and purple glass slides for over 100 years. It relies on very basic chemistry.
Right. Hematoxylin is a basic dye that naturally stains cellular DNA a dark blue or purple color.
And eosin is an acidic dye that stains the proteins in the cell pink. Every hospital does it.
Every single one.
But wait, if human doctors who spend over a decade training their eyes to look at these exact pink and purple slides haven't fully decoded the relationship between the visual shape of the cells and the underlying gene expression.
How could we expect a machine to find a connection?
Exactly. If the humans can't see the RNA in the picture, why should an AI be able to?
Well, what's fascinating here is the limitation of human visual processing versus high-dimensional computational analysis.
Okay, unpack that.
Human pathologists are brilliant at pattern recognition. They look for recognizable architectural structures. The overall size of a gland, the rough shape of a nucleus, how invasive the borders of the tumor look.
The big picture stuff.
Right. But a machine learning algorithm isn't looking at a picture the way we do. It analyzes millions of incredibly subtle pixel patterns simultaneously.
Oh, wow.
It measures the exact mathematical hue of a pink pixel, the microscopic texture of a cell membrane, the geometric relationship between one cell and 50 of its neighbors at the exact same time.
So it is finding deeply hidden mathematical correlations between the image data and the genetic data that the human eye simply didn't evolve to process at scale.
Precisely.
Okay, so just to quickly recap for everyone listening and hit our first bullet point summary here. First, prostate cancer has distinct genetic subtypes like PM50 and PSC that dictate how the disease behaves in response to treatment.
Right.
Second, current genetic testing like the Decipher assay is costly and consumes precious tissue.
It is a huge bottleneck.
And third, the goal here is to use AI to predict these subtypes from standard, cheap H&E biopsy slides.
That's the mission.
So I have an interactive question for you, the listener. If you had to guess right now, do you think an AI looking at a simple picture of a cell could truly predict its underlying genetic code? Hold that thought as we look at how they actually built this thing.
It's a great question. Because the first massive hurdle the researchers had to overcome was purely a data processing problem.
Right, the images themselves.
Yeah, a single digitized biopsy slide isn't like a photo on your smartphone. It is enormous. We are talking about billions of pixels per slide.
Billions.
Yes, it is far too large for even the most powerful AI systems to hold in their active memory all at once.
So to put that in perspective, imagine you are tasked with finding Waldo, but the Where's Waldo book is the size of a football stadium.
Yeah, exactly. You physically cannot take in the entire image in a single glance.
To process it, you'd have to chop the image up into a grid and look at it piece by piece.
And that is precisely what the researchers did. They took these massive whole slide images and chopped them down into tiny digestible tiles, with each tile being exactly 448 by 448 pixels.
Okay.
To train the AI on these tiles, they utilized a framework called multiple instance learning, or MIL.
So sticking with the stadium-sized book analogy, multiple instance learning is essentially handing the AI millions of these chopped up squares and saying, hey, we know Waldo, or in this case, the specific genetic signature, is somewhere in this pile of tiles.
Right.
I need you to look at all of them and figure out which specific squares contain the clues.
Yes, and they tested a couple of different ways to do this. They started by building a baseline model.
Like a control group.
Sort of. This was a standard convolutional neural network, fairly common image processing AI, that they just fed a random sampling of these tiles.
And how did it do?
The performance was, well, acceptable, but not groundbreaking. In medical statistics, we judge these models using a metric called AUC, or area under the curve.
Which goes from 0.5 to 1.0, right?
Exactly. An AUC of 0.5 is basically coin flip, totally random guessing. An AUC of 1.0 means the model is flawlessly perfect. The baseline model they built hit an AUC of 0.718 for predicting the PAM50 subtypes.
I mean, better than a coin flip, sure, but if my life is on the line and I'm deciding on hormone therapy, 0.718 doesn't exactly scream clinical grade reliability? Why wasn't that standard AI good enough?
Because a standard image AI doesn't inherently understand human biology. It's just looking for generic pixel patterns.
It doesn't know what it's looking at.
Right. The researchers realized they needed something much more sophisticated. So they introduced a breakthrough framework they called UniV2-MIL-L.
UniV2-MIL, okay.
This was a massive upgrade because it was a two-stage powerhouse utilizing a foundational model.
Let's define that. The foundational model means the AI isn't starting from scratch on day one of the experiment.
Correct. The researchers used a model called UniV2. Before they ever asked it to look for prostate cancer genetics, this AI had already been pre-trained on millions upon millions of various pathology images.
Oh, wow. So it already had an education.
Yes. It had already learned the fundamental language of human tissue. It already understood what a healthy red blood cell looked like, how a normal duct is structured, and the basic architectural difference between healthy stroma and the chaotic tumor.
It's like teaching someone the rules of grammar and vocabulary before asking them to analyze a Shakespearean sonnet, instead of just handing them the book and hoping they figure out English along the way.
Exactly. That's a perfect analogy. So how did they train this highly educated foundational model to find our specific genetic subtypes?
Yeah. How did they do it?
They used a clever two-step fine-tuning process.
Biff?
In step one, they froze the main UniV2 AI.
Froze it, meaning they locked its core knowledge in place so it wouldn't overwrite what it already knew about tissue.
Exactly. Then, alongside it, they trained a specialized secondary network called a multi-branch self-attention network, or MB-SymAIM-BIAL.
What does a self-attention network actually do in this context?
Its entire job is to look at all those thousands of chopped up tiles from a single slide and learn context. It figures out spatial relationships.
Okay.
And most importantly, it learns to weigh the importance of different areas. It generates these incredibly detailed attention maps, which are essentially heat maps.
Heat maps.
Yeah. These heat maps show exactly which specific tiles the AI thinks are the most highly suspicious or informative for predicting the genetic subtype, and which tiles are just empty space or irrelevant background noise.
So step one is the AI effectively creating its own highly targeted study guide. It highlights only the paragraphs in the textbook that will actually be on the final exam.
Yes. Then comes step two. The researchers took only those top highlighted tiles, the absolute most mathematically informative ones from the heat maps, and fed only those back into the main foundational UniV2 model to fine-tune it.
Oh, I see.
They stripped away all the distracting noise of the massive slide and forced the smartest AI to focus purely on the most critical tissue regions to learn the genetic link.
And when you strip away the noise and let a pre-trained foundational model focus on the highest yield areas, I imagine the results change drastically.
They skyrocketed.
By implementing this specific UniV2-MIL attention framework, the performance for the PAM50 subtypes hit an AUC of 0.863.
Which is huge.
And for the PSC subtypes, it hit 0.81. They successfully pushed the AI past that critical 0.8 threshold into highly accurate, potentially clinically useful territory.
It proves the machine can filter through billions of pixels, locate the most important microscopic real estate, and accurately guess the underlying RNA.
Okay, so let's pause for another quick recap at our 10-minute mark here. Bullet 1. Whole biopsy slides are too large, so the AI breaks them into 448 by 448 pixel tiles.
Yes.
Bullet 2. The researchers used an advanced technique, that MB-SymAIM we mentioned, allowing the AI to weigh the importance of different tiles, focusing its attention only on the most critical tissue regions.
Right, the heat maps.
And bullet 3. This foundational pathology AI achieved highly accurate predictive scores with an AUC over 0.8.
Incredible results.
So here's a question for you listening. Think about how you learn a new subject. Do you scan the whole book or do you find the highlighted summaries? The AI is essentially highlighting its own study guide. What do you think it highlighted?
That is the big question. That leaves a massive glaring question hanging in the air. We know how the AI scans the slides, but what is it actually seeing? When it generates those heat maps and highlights its own study guide, What physical features on the glass slide are signaling a specific genetic code?
Here's where it gets really interesting. This is the crucial bridge between computer science and human biology.
Because the researchers couldn't just accept a black box answer where the AI spits out a score. They needed to understand the morphological physical features the AI was keying in on.
So how did they reverse engineer the AI's brain?
They took all the AI's favorite, highest attention tiles and had the computer group them into 8 distinct visual clusters.
Labeled C1 through C8.
Exactly. Then they brought in a human expert, a genitourinary pathologist, to sit down, look at those clustered tiles, and translate the computer's mathematical groupings into recognizable medical terminology.
This translation phase is where the study gets intensely fascinating. Let's look at the basal subtypes first.
Okay.
This includes the PAM50 basal and the more granular PSC basal neuroendocrine and basal immune types. When the AI confidently predicted a basal genetic signature, the tiles it highlighted were, visually speaking, incredibly angry.
Angry is a good word for it. The pathologist confirmed that these high-attention clusters for basal subtypes were dominated by what we call Gleason pattern 4 and 5.
Okay, for a bit of context, the Gleason grading system is how human doctors score the abnormality of prostate cells.
Right. A Gleason pattern 3 means the glands are still mostly well-formed. They look somewhat like normal tissue.
But pattern 4 means the glands are poorly formed, ragged, and fusing together.
And pattern 5 is total cellular chaos. There's no recognizable gland structure left at all, just sheets of mutated cells invading the tissue.
So the AI correctly and independently linked highly aggressive, chaotic physical structures to the basal genetic signatures.
Yes, but it didn't just look at the overall shape. It zoomed in on something called the NC ratio.
The nuclear to cytoplasmic ratio.
Exactly. The nucleus is the brain of the cell, where the DNA is stored. The cytoplasm is the body of the cell, where the daily functional work gets done.
And in the basal clusters, the AI keyed in on cells where the NC ratio is extremely high.
Meaning the nucleus is massively swollen and the cytoplasm is nearly non-existent.
It's a classic hallmark of a rogue cell. It's dedicating all its energy to rapidly replicating its DNA to divide and spread, rather than maintaining a healthy cell body to do its actual job in the prostate.
Big brain, nobody.
Right. Now, if we look at the exact opposite end of the spectrum, we have the luminal A and luminal differentiated subtypes.
When predicting those luminal A genetics, the AI focused on entirely different visual clusters. These tiles showed much less aggressive tissue. They were mostly Gleason pattern 3.
The pathologist noted the cancer here looked almost atrophic, meaning shrunken with phony glands and a low NC ratio.
The cells appear much calmer, much closer to normal prostate tissue, which perfectly aligns with the generally less aggressive, slower-growing nature of luminal A genetics.
Which brings us to the most complex group, the luminal B and luminal proliferating subtypes. I like to think of luminal B as the tricky middle child of this disease.
It is a notoriously complex subtype, and the AI handled it brilliantly. It clustered the luminal B tiles into an intermediate visual zone. It found a mix of Gleason pattern 3 and 4, but it picked up on incredibly specific nuances that differentiate it from the other types.
Like what?
Well, the AI noticed prominent nucleoli, which are unusually dense, dark spots inside the nucleus itself. It also keyed heavily on a feature called amphiphilic cytoplasm.
This right here is why understanding the basic chemistry of those 100-year-old glass slides matters. We mentioned earlier that hematoxylin is a basic dye that stains blue, and eosin is an acidic dye that stains pink.
Right.
The word amphiphilic essentially means loves both. The cytoplasm in these specific luminal B tumor cells chemically alters so that it absorbs both the acidic and basic dyes,
taking on a very subtle, blended purplish hue.
Wow. The AI was literally detecting micro variations in color chemistry at the pixel level to predict the luminal B RNA signature.
It is remarkable sensitivity. But, the most profound discovery from this cluster analysis wasn't about the color of the tumor cells or the size of their nuclei.
No.
It was about where the AI chose to look when it wasn't looking at the cancer at all.
Right. When the researchers analyzed the attention heat maps, they realized the AI wasn't just assigning high importance to the dense clusters of tumor cells. It was assigning massive predictive weight to the stroma.
The stroma is the connective tissue. It is the blood vessels, the immune cells, the fibrous scaffolding that physically surrounds the tumor.
And for a long time in medicine, we viewed stroma as just the passive background scenery that the cancer happened to be sitting in.
The soil that the seed was planted in.
Yes.
But modern oncology has realized the tumor microenvironment is highly dynamic. Cancer doesn't just sit in the soil. It actively manipulates and corrupts the stroma to help it grow, build new blood vessels, and hide from the immune system.
And the AI, without any human prompting, figured out that this corrupted neighborhood is just as predictive of the tumor's genetic subtype as the tumor itself.
It proved its intelligence by focusing deeply on the connective tissue, a blind spot where human observers historically haven't extracted as much diagnostic value.
So let's recap this section. Bullet 1. The AI's attention maps align perfectly with known human pathology, clustering aggressive cells with basal subtypes and well-differentiated cells with luminal A.
Right.
Bullet 2. It successfully identified nuanced features for the luminal B subtype, like that amphiphilic cytoplasm.
It's a purple U.
And bullet 3. The AI was smart enough to look at the connective tissue, the stroma, not just the tumor cells themselves.
Which is mind-blowing.
It really makes you reflect on the limits of human perception. So, listener, if an AI can teach itself to look at the connective tissue surrounding a tumor to predict its genetics, what other blind spots do you think human observation has in medicine?
It's a profound thought.
But recognizing patterns in a controlled laboratory setting is one thing. The ultimate test of any medical breakthrough is what happens when a real patient is lying in a hospital bed. Does this AI actually change clinical outcomes?
To answer that, the researchers ran the AI's predictions against real-world clinical data. They looked at two distinct, independent groups of patients.
Okay, let's look at the first group. Cohort 2.
Cohort 2 consisted of 131 patients. The researchers wanted to know about hormone therapy responsiveness. Did the genetic subtypes predicted only by the AI actually correlate with which patients got better when treated with hormone therapy?
And the data says yes. Patients whose basic H and E slides were flagged by the AI as having high luminal B and luminal proliferating scores were significantly more responsive to the hormone therapy.
The statistical link was undeniable. The AI predicted PAM50 luminal B score had an odds ratio of 1.51.
Okay, break that down for us.
In clinical terms, that means for every single unit increase in the AI's predicted score, the patient's odds of having a highly favorable response to hormone therapy increased by 51%.
Wow.
The AI was successfully identifying the patients most likely to be cured by this specific drug.
Okay, so if the AI spots luminal B, the oncologist has a green light to use hormone therapy because it's highly likely to work.
Exactly.
But then the researchers looked at the next group of patients, and this is where the biology takes a very sharp, very counterintuitive turn.
Yeah, they analyzed a group of 122 patients, cohort 3, who initially started on active surveillance.
Active surveillance meaning their cancer was deemed slow-moving enough that doctors decided just to watch it closely rather than treat it immediately.
Right. But eventually, for these 122 men, the cancer progressed, and they had to undergo a radical prostatectomy, the surgical removal of the entire prostate.
And after surgery, pathologists examined the removed prostates to see exactly how bad the cancer had become.
And here's the paradox. The exact same patients who had those high AI-predicted luminal B scores, the exact same patients who respond so beautifully and successfully to hormone therapy, also had the highest rates of adverse pathologic features, or APFs, during surgery.
Let's define adverse pathologic features. It means when the surgeon opened them up, the cancer was highly aggressive.
Yeah. It had higher, more dangerous Gleason grades. It was invasive. It had broken out of the prostate capsule and invaded the seminal vesicles, or even spread outward into the regional lymph nodes.
Wait, I need to stop you there, because that feels completely contradictory. So what does this all mean? If luminal B is this highly invasive, rapidly spreading aggressive cancer, how is it simultaneously the best candidate for hormone therapy?
It sounds impossible, right?
Yeah. How is a high score a good thing for treatment if the cancer itself is so dangerous?
It is a brilliant biological paradox. And if we connect this to the bigger picture, it reveals a profound clinical duality about how this disease operates.
I'm listening.
Luminal B and luminal proliferating tumors are highly proliferative. They are fast-growing, aggressive, and highly dangerous, which is why they display those adverse surgical features. They are massive engines running at absolute maximum capacity.
And what fuels a massive engine?
A massive amount of fuel, in this case, testosterone. Because these specific cancer cells are multiplying so aggressively, they become incredibly disproportionately dependent on androgen signaling.
They are addicted to testosterone to sustain their rapid growth.
Yes. So what happens when an oncologist administers hormone therapy and suddenly completely cuts off that testosterone supply?
The engine completely starves. It's running too hot to survive without fuel, so it crashes faster and harder than a slower-growing tumor would.
Exactly. The very mechanism that makes a luminal B tumor so dangerously aggressive is the exact same mechanism that serves as its Achilles heel.
That makes perfect sense. And the truly stunning part of this study is that the artificial intelligence successfully captured this entire complex biological duality.
It predicted both the intense danger of the tumor's adverse features and the highly successful cure rate of the hormone therapy, purely by analyzing the digitized pixels of a basic pink and blue glass slide.
It found the invisible genetic truth without ever needing the $3,000 genomic test.
Exactly.
So, summarizing this final block here. Bullet 1. High AI predicted luminal B and luminal proliferating scores strongly correlate with successful hormone therapy.
Right.
Bullet 2. Paradoxically, these same subtypes correlate with aggressive adverse pathologic features.
Because it's an engine running hot.
And bullet 3. The AI successfully captures this complex biological duality, highly aggressive, yet highly treatable with specific therapies without needing the expensive genetic tests.
No, obviously the researchers are careful to note that this is a proof of concept.
Right, it's not in hospitals tomorrow.
No, the AI needs larger prospective validation trials across diverse hospital systems before it officially replaces a test like the Decipher assay.
But the foundation has definitively been laid. And the implications of that foundation are staggering. We're talking about fundamentally reshaping global health equity.
It transforms a qualitative, subjective visual assessment into a deeply quantitative molecular insight. It effectively expands the diagnostic toolkit of every pathologist on the globe, regardless of their clinic's funding.
If this technology scales, it means a small, rural clinic anywhere in the world, provided they have a basic microscope to make an H&E slide, a digital scanner, and an internet connection,
could one day offer the exact same highly personalized genetic-level prostate cancer profiling as an elite research hospital in a major city.
We're turning a century-old dyeing technique into cutting-edge DNA data, which leaves us with one final provocative thought for you to carry with you today.
Yeah.
We've just explored how an algorithm can extract an invisible genetic signature from a piece of glass by looking at the connective tissue and the hue of a pixel. It forces you to ask, what other invisible biological truths are currently hiding in plain sight?
It really does make you wonder.
What massive medical secrets are locked inside our most routine, everyday diagnostics, like a standard blood draw, a basic EKG heartbeat tracing, or a routine chest X-ray, just waiting for the right artificial mind to finally decode them?
The possibilities are endless.
Thank you so much for joining us on this deep dive. We deeply appreciate your curiosity and for choosing to spend your time learning and exploring the future of medicine with us today. Keep questioning the world around you, and we'll see you next time.